And so by now we all, I think, appreciate the incredibly crucial roles that microbes play in eukaryotic evolution and health and development. But understanding the processes that lead to these symbioses, how they evolve, is much more challenging. Specifically, we'd like to know what kind of microbial phenotypes are targeted by host selection and how microbial genomes overcome their own constraints and navigate the trade-offs that are implicit in adaptation. And generally speaking, are there general principles, universal principles that guide symbiotic adaptation? And so we're going to address these questions using this bobtail squid host to experimentally evolve a bioluminescent symbiont called Vibrio fisheri. And what I'm going to show you today is that in doing this, we found that the host repeatedly, consistently exerted very hard selection to select on variants in a key global regulator across multiple strains. And in doing so, these variants seem to unlock, through pleiotropy, multiple symbiotic traits. All right, so Vibrio fisheri is the bacterium in question. And it's an awesome system for addressing evolution of symbioses because it's incredibly widespread and very ecologically diverse. So there are a number of free living strains, but there are also tons of strains that form these very specific and intimate associations with animals like fish and squid. And what's really interesting about this ecological diversity is it doesn't seem to be informed by phylogeny. And so when we do a whole genome phylogeny, we see these different ecotypes sort of distributed across. And this suggests that this host specialization, this symbiosis, has potentially evolved several times. And further, when we use our little host bobtail squid to assess the symbiotic capacity of these various strains, what we see is that neither geography nor genetic relatedness seem to dictate how capable any given strain is to form a symbiosis. So this strain right here, for example, H905, is found in the exact same seawater environment as the host native strain, ES114, and yet it takes way more cells to successfully infect or colonize the squid. And so what this suggests is potentially any one of these strains could readily adapt to symbiosis, given strong enough selection. And so what's in it for these two partners? So for the squid, the benefit is camouflage, protection. So it spends most of its day buried in the sand, but at night it goes up to forage and has to deal with avoiding its own predators. And to do this, it uses the light produced by the bioluminescent bacteria to effectively mask its own silhouette against the moonlight. And this is a really sweet deal for the microbes too. So the squid are born sterile, but within minutes of hatching, these little ciliated arms and the surface of the leg organs start waving around and recruiting Vibrio fisheri from the surrounding seawater. And within hours after colonizing, and these leg organs can be colonized by fewer than 10 cells, we think, very small founder sizes. Within hours, they're approaching capacity, they're bioluminescing, and by sunset or sunrise the next day, the host is venting out over 95% of the population. And it's going to do this regular bottleneck every single day for the rest of its life. And so as you might predict, the waters in these squid habitats are greatly enriched for symbiotic fisheri. So we capitalize on this sort of natural bottlenecking feature of the host to evolve strains that had various symbiotic capabilities to see if we can improve their ability to form an association. And so to do this, we grew up an ancestral inoculum for different strains. We exposed them to squid, and squid that got colonized, we let the population grow up and vent out and regrow, vent out, and we take that ventate and we present it to the next squid in a serial passage format. And we continue doing this for about 360 bacterial generations, and then we sequenced the genomes from different temporal or different time points along this process to see what happened. And we did this for multiple strains, and we did multiple replicate lineages for each strain. And so basically what we found was truly surprising. First of all, I just want to point out that, so each of these strains, each line here represents the consensus genome from a replicated evolutionary lineage. And you can see right away that there are a couple strains here, including the native strain that we evolved as like a control, didn't accrue any mutations. And so what's interesting about these is they were already very good, or relatively good symbiotic partners, and so they really didn't stand to improve much, at least within 300 generations. However, our worst strains, originally a fish strain called MJ11 and a planktonic strain called H905, independently in their replicated lineages, picked up different variants in the same locus that at the time was uncharacterized, and we are calling REM-S. And I'll just focus on MJ11 for the remainder of this talk. But what's really interesting about all these independent mutations that accrued, they're all non-synonymous and they're all in extremely conserved domains, suggesting they could have a really big impact on protein function. But are they adaptive? Well, yes, they are. So we see that strains harboring these various mutations are all vastly better at colonizing squid compared to the ancestor, and also as a control, we evolved the same strain in culture, traditional passaging in culture, and we can see that the squid evolved variants fare way better in the squid. And it's important to note that they do not confer any general rigor. So this benefit seems really exclusive to the environment of the host. So just how strong of selection is this host exerting? And so to do this, we borrowed this model that assesses the survival probability of beneficial alleles that are facing repeated bottlenecks, so very similar to the squid system. And by plugging in the salient parameters from the squid system, what we noticed right away is that even alleles that confer relatively strong selective advantages, like a selective coefficient of 2 is pretty high, these variants do not fare very well unless they originate very early in inoculum growth or very soon following colonization. And what was really interesting about this model is that its variants arose after the very first venting cycle and in the subsequent venting cycles. Their selective coefficient didn't need to be as large to survive. And so this made us wonder, we already saw from the previous graph that these symbionts or these variants are very good at successfully colonizing the host. But maybe they're also good at persisting during the subsequent venting stages. And so to do this, we did a head-to-head competition of ancestor and evolved symbiont in a squid, and we measured their growth rates after 24 and 48 hours. And what we see is that in these co-colonized squid, that the selective advantage nearly doubles after 48 hours, which implies that in addition to their improved ability to initiate colonization with the host, they're actually growing better and faring much better in the face of these host-imposed venting bottlenecks. And I'll just point out that we think this is a case of hard selection because the selection coefficients don't seem to have any relationship to starting ratio or density. Okay, so very quickly, how could these single mutations possibly confer this improved, vastly improved symbiotic ability? And so here are a number of features of the squid-like organ that we think are affected by these mutations. So foremost, what we see when we do these phenotypic assays is that our evolved allele, which we're calling REM-S1, produces way more biofilm than the wild type and the ancestor. And I won't go into the details of all this, but we basically figured out that in the wild type and the ancestral form, this protein acts to repress biofilm production, and so these mutations seem to alleviate that repression. And as a consequence, we think that this extra polysaccharide production is facilitating this improved aggregation ability. So recall when they colonize the light organ, shortly after the squid hatches, they have to form an aggregate in the mucus, in the squid's mucus, and that aggregate is facilitated by the polysaccharide, and then that aggregate can migrate into the interior and begin to grow. We also think that this polysaccharide is improving their ability to resist oxidative stress, which is the real hallmark of the interior environment of the light organ. What was really cool and unexpected is that this improved biofilm ability seems to confer an advantage of avoiding the host's immune defenses. So in addition to the oxidative stress, which is a method of defense, the squid-like organ is patrolled constantly by these macrophages. And what we see is that in the ancestor, when we subject the ancestral cells to macrophages, they are frequently bound up and phagocytosed. Whereas with one single mutation, the evolved symbiont is bound. It's reduced its binding to a level very comparable to the native symbiont. So in addition to these traits, which we believe are all related to the increased polysaccharide production, we also see reduced luminescence, which is a trait that we know the squid is very particular about. There's lots of examples of how the squid can tailor the light production in the host. And then, as if this wasn't enough, when we do like wholesale transcriptomic and metabolomic or metabolic profiling, we actually find quite an amazing shift. So the center columns are the evolved allele, and you can see there are a number of genes that are very similar to the ancestor, but there are a number of genes whose expression really changes quite dramatically. And obviously I don't have time to go into all of these details, but please ask me after. It's pretty interesting how this one regulator can really dramatically change the physiology of the cell. All right, so to sum up, what we found is that this strong selection imposed by the host has favored these mutations repeatedly in the same sensor, which is the regulator. I did this very rapidly in the course of the evolution experiment. But the strength of the selection varied temporally, and that the single mutation was able to improve multiple symbiotic traits simultaneously. With that, I'll take any questions.